How are you? Are you tired? No, not at all. Yeah. I am sorry, guys. I could not join earlier.
But evening, you will come. There will be a farewell party.
Sure, I'm first there. Yeah. No, just I came back from another meeting, so I could not attend the previous parts of your program. Hopefully you enjoyed that. And just one last bit of patience. Okay. So I put the title today is Ultrafast two-dimensional spectroscopy. So, and the second line is the photosynthetic light harvesting, and I added complexes here just to finish the sentence. Now this talk is as much about spectroscopy or lasers or whatever as it is about biology. Because I'll go back one slide, I'm here from Szeged. We do mostly biology here, and we are in what's called Institute of Plant Biology. And the spectroscopy part actually is not done here. We don't have the 2D spectroscopy in Szeged yet, but we are doing that in collaboration currently with Singapore and with Nanyang Technological University. And we do other kinds of spectroscopies here, but I won't talk about that much today. So, as I said, it's, I will say, maybe half of the time I will spend on biology and more specifically on photosynthesis. So, probably not many of you are in the field of photosynthesis. Yeah, I can see the faces. Okay, and that's why I will start with some refreshment or refreshing crash course in photosynthesis, and then talk about the method, so that finally I can tell you some of the results that we have as a case study. I'm not going to do a very extensive review on the 2D spectroscopy and its all possible applications. There is plenty of literature on that. There are books and review articles, because the 2D spectroscopy nowadays is very hot. Many labs are doing that, and basically I can say from what I can see in the last 2-3 years, it is actually replacing the standard ultra-fast spectroscopy techniques like transient absorption. So whoever was doing transient absorption is now just doing two-dimensional spectroscopy. And as you can see, it's not a big deal. One is just extension of the other. But as I said, I'm not going to give you a review on that. I'll just tell you as an example, as a case study of what we are doing with this technique and what we can get from it. And then maybe after 2-3 hours I can tell about the 3D spectroscopy, which is another extension of the 2D spectroscopy. Maybe 5 years from now everybody will do 3D spectroscopy instead of 2D, and then 4D. So, like Gillette, every time put another knife, another blade in the laser, which is good. So, photosynthesis. Why do we bother to study photosynthesis? And I must say also, it's not an exaggeration. This field is also gaining a lot of attention and traction nowadays of photosynthesis research. So why is that? This is because photosynthesis takes something bad and gives something good, right? It basically takes bad stuff away from the air, puts good stuff in the air, and in the meantime produces what? Sugar. Yeah? And all this just by using sunlight. So that's photosynthesis. The production of carbohydrates from carbon dioxide and water using the energy of light. And hello? in the atmosphere, which is very good if you can breathe, and also all the biomass, all the fuel and all the food for us to eat and burn. And maybe there is no need to explain this graph, because it is also a very hot topic nowadays. Even the Pope is on board. This is, of course, the correlation plot of the CO2 concentration in the atmosphere and global temperature anomaly, that is change in average temperatures over time in the last century. And you can do the conclusions yourself. No need to mention them. So photosynthesis is very important, and actually it is one of the, maybe understated, it is one of the best tools that we have to tackle climate change and to tackle our carbon emissions. Just plant more trees. And so they will remove the CO2 from the atmosphere and counteract the climate change. And at the same time, we can have, by using photosynthesis, a clean and renewable energy source. And this, for this, it's very good to put some things in scale. This is the global solar power heating the Earth's atmosphere at any moment in time, on average. That is 173,000 terawatts. Globally, photosynthesis, that means all the trees and all the algae in the oceans, they use 120 terawatt power, which is what 0.1% of the solar energy coming on the Earth. And we need 10% of that. So if we can get 0.01%, we are done. We don't need to be worrying about energy anymore. And that is not so much compared to that. So the worry is how to make use of that energy. And that's what all the efforts are about nowadays. Try to understand the physical, biophysical, fundamental underpinnings of the photosynthetic process so that we can make use of it, combining it with our engineering and so on, skills to make artificial devices and so on, whatnot. But we are talking now about the nature of photosynthesis. So I cannot see my slides. So that was the wrong way. Yeah. So what is that? This is the chloroplast, which is where all the photosynthesis in the green plants take place. The green plants that also include all the algae, anything that is green or has oxygenic photosynthesis, except the cyanobacteria. They don't have chloroplasts, but the chloroplast is actually a cyanobacterium which entered someday into another cell and started to do photosynthesis there, which is called endosymbiosis. So this is the chloroplast, and what it has is it has a double membrane envelope, outer and inner envelope, and inside what you see, these discs, what are those? Those are the thylakoid membranes, the discs or the sacs are called thylakoids, and they are connected to each other with stroma, lamellae. These are membranes. These are very unique membranes. This is basically one continuous membrane that separates the volume of the chloroplast in two electrically isolated parts, which is one is called the stroma, and outside and inside this vesicles is called the lumen. Electrically isolated because this is, of course, the plant solar panel, and it makes the charge separation and the electric potential, or actually electrochemical potential across that membrane. So there's also another separation, functional separation, let's say, because there are two types of reactions in photosynthesis, if you remember from school: light reactions and dark reactions. Light reactions, what is that? That is the biophysical and photochemical part of the process. That is using the light energy, electrons are extracted from water, oxygen is released in the atmosphere, electrons and protons are used to create ATP, which is the fuel of the cell, and NADPH, which is the, let's say, electron carrier. And these two are then used in the dark in the Calvin-Benson cycle that takes the carbon dioxide from the atmosphere, uses or from the water, uses the energy in the ATP and the electrons to reduce the carbon dioxide into sugar. And so these whole light reactions, of course light-dependent reactions, happen on the membrane or inside the membrane, and the dark reactions happen in the stroma, so outside here in the space between the membranes, right? So that is where things happen. Now we are from now on forgetting the dark reactions, only going to the light-dependent reactions. The light-dependent reactions, what happens? Light is absorbed by what we call photosystems, the photoactive pigment, which is what? Chlorophyll. And chlorophyll is excited. The redox potential is enough actually to take the oxidized chlorophyll has enough oxidizing power to take the electron from water, and the excited chlorophyll loses one electron, the electron goes through the chain of electron carriers, reaches the other photosystem, photosystem 1, where another photon is absorbed, another photoactive pigment makes the same, loses the electron, and the electron finally goes to NADPH. So that's the linear electron flow from water to NADPH. The job is done. And this is the same thing, just in this scheme we also see the actual structures of the photosystems. That's the photosystem 2, that's the photosystem 1. These are crystal structures, X-ray crystallographic structures of the whole super complex of photosystem 2 and 1, and the cytochrome which is in between, and some other stuff, and also the ATP synthase. All these are crystal structures. And this is the electron flow, of course, as I said, from water through cytochrome to photosystem 1, and then finally via the ferredoxin NADP reductase, one enzyme that reduces NADP to NADPH. So all the electron carriers and all the photoactive pigments are bound to proteins or protein complexes, or pigment protein complexes. Now here one very important part is missing actually in this picture. This is what is missing is the so-called antenna proteins, the light harvesting antenna. Now the thing is, one chlorophyll is very small. It cannot, and in natural conditions, not in laser light, but in natural sunlight, there are not so many photons around. So all photosynthetic organisms, without any exception, have a way to concentrate the solar energy onto the photoactive pigment, to funnel the energy to one pigment that will do all the photochemistry. Because photochemistry is expensive for the cell to build. It's bioenergetically expensive. You need to build all those proteins to contain the cofactors, the electron carriers, and this costs. Protein is expensive. You cannot just build so many. So that's why all photosynthetic organisms have antenna. That means a protein, usually, not always a protein, but usually, that contains a lot of chlorophylls. A lot of chlorophylls, they can collect a lot of photons, but they don't do photochemistry. So not each chlorophyll has all the reaction systems bound to it. So these are the light harvesting antennas. So what happens is that any chlorophyll can absorb a photon of light, and then the light will be transferred. The light energy will be transferred from one to another, more or less like a random walk, but not exactly, towards the special pigment, the special chlorophyll that will actually do photochemistry and which has all the machinery necessary to complete the photosynthetic reaction. So, as I said, it's not exactly a random walk because energy is normally transferred downhill. What does that mean? It means pigment excited states have a different energy, transition energy. So because of that transition energy, they will of course absorb certain wavelength or frequency of radiation of light, and then the excited state will be transferred to another pigment which usually will have smaller transition energy, small or lower energy excited state, in other words. So that's why that way, let's say if we have we don't have really rainbows. Mostly everything is green, but not only because the chlorophyll also absorbing blue, and there are carotenoids in between, but that's another story. So let's say in this rainbow picture, if a blue pigment absorbs light, blue absorbing, it will not look blue. Blue absorbing pigment will transfer to green absorbing pigment and so on, and usually the reaction center is the one that has the lowest energy. But it must still be high enough to drive the inoxygenic photosynthesis, to drive the oxidation of water. So we have a limit here on the transition energy of the photoactive pigment. And actually the antennas are very different among all the organisms that you can find, but the reaction centers are remarkably the same. For 2 billion years they practically didn't change. Right. So anything else here? Nothing else. Okay. How does the energy transfer work? Probably many of you know the first mechanism, because it's very commonly used in microscopy and spectroscopy and in all kind of lab sciences. So the first transfer is also called resonance or induction resonance transfer. Basically it says that you have one box is one molecule, another box is another molecule, and these are electrons on these two molecules. One molecule we call donor, the other acceptor. We have an excited donor, that means one of the electrons is on a higher level or higher orbital, if you wish to use classical terminology, higher orbital, and because of the dipole-dipole interaction between the two, somehow the excited state of that molecule will induce the transition in the other molecule, or it will induce dipole interactions are very similar in a classical representation as coupled oscillators. So if one spring oscillator starts to vibrate, oscillate, the oscillation will be transferred to the other. That's the induction, the resonance induction. And as a result, this will lose the energy, this will gain the energy, and in the end, the other pigments will be excited. And then there will be another pigment here, and another, and the last one somewhere here will be the reaction center. Okay. So this is all very well elaborated, first in classical, also in quantum mechanical terms. It depends on the distance between the pigments, it depends on that's very important here, sixth power of the distance. It depends on the spectral properties, that's the spectral overlap between the two, which is also the resonance condition. It depends on the orientation of the two molecules. So this always takes place in the in the antenna pigment protein complexes. But unfortunately, that's not the whole picture. This theory has a lot of limitations, and the limitations are that the pigments are basically not too strongly coupled to each other. Because if they're too strongly coupled to each other, then they start to behave like a supermolecule, or what is called. We have exciton delocalization of the excited state, and then this theory doesn't really work. So there is exciton theory that can predict the energy transfer in the strongly coupled case, but there is not a really good theory in the middle when the pigments are neither strongly coupled nor weakly coupled, but intermediate regime. And all the first things, the pigment protein complexes are exactly in that intermediate regime, where it's very difficult to calculate and model energy transfer. Okay, that's theory, and we're talking about experiments. So here are some structures, more structures, that again for system 2, and I just want to show you the antenna. This is the core complex. The core complex is where the photochemistry takes place in the reaction center. This is a dimeric core complex, a dimeric core, which has one reaction center somewhere here, one somewhere here. All other pigments are antenna. These are core antenna, and this is peripheral antenna, or what is called light harvesting complex, LHC. So we have here, this is a LHC, this is LHC, these are actually 3 LHCs, or LHC trimer. And this is the one that we in SEGLET, in our group, mostly studied. The major light harvesting complex 2, which is actually the most abundant protein or membrane protein in the world. And this is again the same protein, just from a side view, just to show you. So that gray part is the apoprotein, the protein itself. It has these alpha helices which span the membrane. Here is the membrane, and here is the membrane. It is a transmembrane protein, and it binds a number of chlorophylls, the Greek stuff, and a number of carotenoids, which are very important molecules to protect. Also, as they are protecting people, they also protect the light harvesting complex from photo damage and from oxidative damage. So, as I told you before, there are a bunch of chlorophylls whose only function is to collect more light, transfer it between each other, and finally it must go to the reaction center. Okay. Now, all these chlorophylls are the same. They have the same chemical structure, but they have different energies. How does that happen? This happens because the protein tunes the spectral properties of the pigments. Because of electrostatic interactions, the environment of every pigment is slightly different. So we have actually an energy funnel even in this protein, but the energy funnel is not very clear, because if you look at the structure, you only see identical pigments. That's not exactly true. 2 types of identical pigments, chlorophyll A and chlorophyll B. So they are chemically different. But chlorophyll B has higher energy, chlorophyll A has lower energy, but we have 6 of those and 8 of those, and we don't know in between how the energy goes. And we would like to know. This is the same protein, but now only the chlorophylls, and they are mostly coded by their energy, but this is very rough. So blue is the highest energy chlorophyll B's, and red are the lowest energy chlorophyll A's. This we know, but in between is a bit of a guesswork. And the energy transfer goes from, usually we can tell that, okay, these are very close together, there will be fast transfer between them. These from here to here maybe slower. We can guess, we can use Fjordster theory or whatever to make predictions, but it is not so easy to understand how the energy goes in this complex, just from theoretical point of view. There have been no shortage of models. There are plenty of calculations, theoretical calculations. This is one example from one of our collaborators in Austria, who has calculated the energy transfer from between different pigments using one theory. But the thing is, as many theories there are, as many models there are. Every model has a different solution, and the rate constants, this rate constants in energy transfer can vary by more than an order of magnitude. So clearly, theory is not good enough. We need to actually measure these rates. And these rates, as you can see, they start from 10 to the minus 14. That's very fast. That's 100 femtoseconds. So that's why we need ultra-fast spectroscopy if we want to understand anything about energy transfer in such systems. However, just by having ultrafast spectroscopy doesn't give you much, because, as I said, all these pigments are green, and that means it's very difficult to tell them apart spectrally. They are very similar, and that's why we need ultrafast, high spectral resolution methods. Right. Okay, enough about biology. Let's talk about spectroscopy now. Do you know what is spectroscopy? And do you know what it means? What means spectrum?
Spectrum.
Yeah. It means a ghost. Yeah. Yeah, it's the Greek word for ghost. And spectroscopy means seeing ghosts. That's right. Because we actually see the ghost of the molecule. We cannot see the molecule with chlorophyll. We can only see the effect that the molecule has on the light that we pass through it. That's spectroscopy. So, but what is transient absorption spectroscopy? Now you take over. So, just a few words. I will tell you about the principle in very, very rough terms, because our goal is to go into two-dimensional, but it's, as I said, it's just an extension of this classical pump probe spectroscopy that is so well known and so widely used for decades now, and very well elaborated theoretically and in practice. And there is in this university very good basis and technical, as you know already. So, in pump probe spectroscopy, we want to follow the dynamics of the system in time. And that means we perturb the system, and then we follow the changes in time. That's why it's transient. But it's absorption spectroscopy because we measure and quantitate the absorption of light by our material specimen. And it's pump probe because we use two pulses of light, that one we call pump and the other we call probe. So what is the pump, what is the probe? The first pulse hopefully will be absorbed by some subset of our molecules, and they will get into the excited state, and then we will give them time to evolve via their natural processes. So the excited states to be converted to whatever product states they will do. Chemistry will take place, maybe, and then after a while we will send another pulse to see what has happened during the time between the pump and the probe. Right, that's the basis. Flash photolysis, or pump probe spectroscopy, or it has many names. Now, what we actually measure is, in pump probe spectroscopy, is the differential absorption. So we don't measure the absorption of this pulse, we measure the absorption that occurs, the absorption of this light, which is the probe light. And we measure the difference between the absorption that occurs if we did not have the pump minus the absorption that is, or the other way around. The absorption that occurs after we have the pump, and we subtract from it the absorption that happens when we did not have the pump. Here it is. So this is differential absorption signal, and mostly what we see in transient absorption as a signal is 2 or 3 signals. One is called ground state bleaching. Ground state bleaching is because let's say if we send the pump pulse, and some of our molecules absorb and they are in the excited state, if the probe afterwards overlaps with the pump frequency, we will have smaller concentration of molecules that can absorb, right? Because some of them are already in the excited state, they cannot absorb yet. So that's called ground state bleaching. We will see negative absorption in the difference. The other thing is, if we have overlap of the probe and the pump pulse, and we have some excited state, we also, of course, have a probability to induce what? Emission, stimulated emission from the excited state back to the ground state. Emission is also a negative absorption, so this is another negative signal that we observe. We can also have, actually, what this shows is the third signal. This is the excited state absorption. So it means the second pulse found the product states. In this case, this is first electronic excited state, and this state can absorb. So this will give positive absorption. That will be the third type of signal, mostly that we can follow. And hopefully the gap, or the energy gap between the first and the second levels will be different. Then we will see in this case, if this gap is different to this gap, then we will see the bleach signal and the excited state absorption signal in different frequencies. Okay. For this, of course, we need a broad pulse that has a lot of frequencies. So this is then brings to the very most common experimental setup in transient absorption is the narrow pump, broad probe. That means the pump pulses have narrow bandwidth. Then we can select a specific subset of chlorophylls, let's say, to be excited, and then with a broadband probe, we can see where the energy went, which other molecules hopefully got excited after some time, or what other products we can generate. And fairly simple to draw it, not so simple to build it on the table, but the also very classical setup for transient absorption. We need to start with a pulsed laser, obviously, and hopefully that pulsed laser will be a mode-locked, ultra-short pulsed laser, like a titanium sapphire oscillator. Usually we will have also, if you follow the technical parts, we will have amplifier or another stage, laser stage, to amplify the emission, the laser emission, so that we can then do a lot of nonlinear manipulation, nonlinear transformations of the light that comes originally from the laser, so that we can, for example, use an optical parametric generation. That is, people use optic parametric oscillators or optical parametric amplifiers. They're slightly different devices, basically another laser, to make the narrow band pump frequency that we want, so that we can excite exactly the molecules that we want. And then we send this beam to the sample. Then another branch of our spectrometer, here this is a beam splitter, and we usually take a much smaller energy part of the original fundamental laser, and from this we generate the broadband probe pulses. What says here white light generation, this is usually a crystal that's commonly sapphire, that will generate white light or very broad pulses in frequency. And then we match these two pulses on the sample. One more thing here is the mechanical chopper we use. Why? What does it do? It basically cuts every other pulse so that we can have the difference of plus and minus after pump and without pump. Okay? And the other most important part is that we must delay one of the pulses so that we give some time to the system to evolve, and this delay will be variable delay. So we will use a variable delay stage to increase the path of the light so that it arrives slightly with a little delay to the sample, and then we will scan over some range of delays to cover the time range where the processes that we're interested occur. Let's say from femtoseconds to picoseconds, can be nanoseconds, millisecond, whatever processes we are looking at. Finally, what happens, as I said, we measure how much of the probe signal arrives at the detector. But for the detector, normally, if we use broadband pulses, that means we are doing multicolor experiments. That means there are many frequencies in our pulse, and we want to frequency resolve them, so we use a spectrometer. There's a grating or something like a monochromator in the normal, or half monochromator, if you wish, in the normal absorption spectrometer that you have. And of course there is a detector and a computer, and that's it. So what do we get? We get differential absorption as a function of frequency and as a function of time. So, 2 dimensions, you will say. Is this 2-dimensional spectroscopy? No. This is 1-dimensional spectroscopy. Yes. So, because some people, when they see two-dimensional or 3-dimensional, whatever plot, that doesn't mean this is 2-dimensional spectroscopy. It's 1-dimensional because we have one spectral or frequency dimension. This is here wavelength. Actually, this is an example, I forgot to put a reference to show off. An example of another type of antenna complexes, which I did not show you, just to confuse. These are bacterial antenna complexes, slightly different from the LHC1, from the LHC2, but it's the idea is the same. I just want to show you the kind of signal that you can get with a light harvesting complex with a femtosecond transient absorption. We have here reconstituted in vitro 2 types of complexes. One is the natural complex as it is or similar to the native one. And the other is basically we destroyed its antenna functions by putting a molecule that will dissipate all the energy to the ground state. So if the antenna must function as antenna, it must keep the excited states long enough so that they reach the reaction center. And if we put a quencher, what we call, or a molecule that will dissipate the excited state, then this cannot be an antenna anymore because the energy cannot reach the reaction center, it will just be lost. So it's like a radiator that kills the excited state energy. And then if we measure the transient absorption signal, you can see in these normal active complexes, here this is the time and this is frequency dimension or wavelength. At 0 time the pump pulse arrives and we have signal. And then nothing happens. Over 100 picoseconds. Very slowly the energy will be lost via non-radiative and radiative decay routes. And here in this dissipating sample, we hit it with the pump laser and then immediately, not immediately, but very soon, it will decay, we will lose the signal. So this signal actually is negative, as you can see. This is the ground state bleaching of in this case, bacterial chlorophyll. And here we can see positive signal, that is the excited state absorption. You can see the same here, but everything is lost very quickly. So, that's another example now with chlorophyll in LHC2. And what we have here is the normal linear absorption spectrum of LHC2. LHC2, as I said, it has two types of chlorophyll, B and A. B absorbs here, A absorbs here, lower energy. These are calculated, theoretically calculated energy levels of chlorophyll B and chlorophyll A in the complex. And here is our narrow pump. And then we use a broadband to cover all the states. So we excite chlorophyll B, and then we probe over the whole region, and we can see here spectra at different delay times. So in the beginning, let's say 300 femtoseconds after the pump, we have a negative signal. That means bleach. That means there are some chlorophyll B's excited, and there are also some chlorophyll A's excited. Then when time progresses, we see the decrease of the chlorophyll B band, and at the same time, the chlorophyll A band increases. So what we see is energy transfer from chlorophyll B to chlorophyll A. And we can take this point and take this point and plot them in time. So these are the time traces will show, again, 0 is here, this is negative signal. The chlorophyll B signal will decay, and the chlorophyll A signal, here will rise at the same time. So this is the energy transfer. But not much spectral resolution we can get here. Because all the chlorophyll A's are here, all of these are here, we cannot tell them apart. There comes finally the two-dimensional spectroscopy to help us with this problem that we cannot actually get so much information from the normal transient absorption because we cannot resolve our pigments. Now, a little help we get from adding another frequency dimension. So, here we have resolved in frequency our probe light, or we can tell where the energy is at some moment in time. But what we can't tell is where the energy came from. And the two-dimensional spectroscopy will tell us where the energy is and where it came from. I will explain that in a minute.